Welcome to the Market Bull podcast, where we dive into the stories shaping Australia's finance, tech, and resources landscape. Each episode, we will be sitting down with ASX-listed executives, industry experts, emerging explorers, and innovators to unpack the latest trends driving investor attention. Whether you're a retail investor, market watcher, or just simply curious about the companies making headlines, this is your place to hear Australia's next generation of growth stories. I'm Matthew Craig, and this is the Market Bull podcast, powered by themarketbull.com.au, your home for small cap coverage. Hi, I'm Matthew Craig, and joining me online today is the Imagion Biosystems Chief Business Officer, Ward Detwiler, and the Co-Director of MRI Research at Wayne State University, Dr. Mark Haacke. Welcome, guys.
Hi, Matthew. Thanks for having us.
Thank you, Matthew.
So maybe I'll just start with you, Ward, just to introduce us and give us a reminder of the Magsense technology and just what Imagion Biosystems is working on and, why we're here today.
Yeah, absolutely. So Imagion is a clinical stage medical imaging company really focused on changing how we detect and diagnose cancer. We're doing that through our Magsense imaging agent technology, which is a first of its kind targeted magnetic nanoparticle, which brings molecular specificity to the over 50,000 installed MRI systems globally. Our first product focused on this improved staging of HER2-positive breast cancer has successfully completed its phase 1 trial, and we are now preparing an IND submission for our phase 2 study, which we expect to submit later this year. But what I think is really most unique about Magsense beyond the HER2 implication is that this is really a platform technology. While we are laser focused on our HER2 and completing the phase 2 successfully, we have additional applications in prostate and ovarian cancer that have completed a lot of their preclinical work and are now ready to be prepared for IND enabling studies in the future. So that's something that we're keeping our eye on. And pretty much anything that we can find a targeting ligand to address, we can find a cancer that we may be able to go after and help improve the diagnosis of.
Yeah, awesome. And just over to you, Mark, maybe just give us a bit of a background on, how you've got here today, all the work that you've been doing, and then just maybe give us an overview of why molecular MRI is so important.
Thank you, Matthew. Well, I've been doing MRI for 40 years now. MR imaging is really an exciting field. It has advanced tremendously, as you can imagine, over 40 years in terms of the equipment itself and many new developments in both hardware and software in MR imaging, as well as many new technical advances in how we're able to probe pathophysiological processes in the human body. And although a lot of MR today is dominantly structural MRI, it's also possible to do things like image the blood vessels of the human body. So if you are delivering a drug through the vascular system, for example, we can map that vascular system out very accurately. And along those lines, we have actually used something called ferromoxetol, which is an iron-based contrast agent to image the vasculature down to 50 microns. So when we have an iron-based substance, it makes it possible to dramatically improve the detectability of small structures. And many years ago, I think back around 2017, I published a paper using, again, ferritin to see, how accurately could we measure iron content. And I believe we measured iron down to the level of 6 picograms of iron per cell. And we also showed that we could image with ferritin, which is, I think, similar to the iron-based contrast that Imagion is using, that we could literally image a few hundred cells. So that's very impressive what MR can do. Of course, those were test tube measurements. Now we have done something similar to that with the current doses that we were given by Imagion, down to now the level of about 1 microgram per milliliter in terms of the dose. And we can still see that effect quite well. So having that ability to peg these type of changes in the body, especially cellular changes occurring in cancer, for example, makes it possible to really search and tag very specific pathophysiological processes. And I think that's really the future for MR. Can we design other technologies? And I don't think anything is more powerful than iron-tagged technologies to continue to enhance our ability to use MRI, not just for structural imaging, but for molecular imaging at the level that we're talking about here with Imagion. So can we run this for all of these 50,000 magnets Ward's talking about? Well, the ability to do this is certainly best at high field. And by high field here, I mean maybe 1.5T and 3T, where the presence of a magnetic contrast agent basically enhances our ability to distinguish these small structures. And although I'm not sure how many that is out of the 50,000, but even if it's half of that, it's certainly a lot of machines worldwide. And most of the modern scanners today are high field scanners. So that opens the door for not only imaging these pathophysiological processes, but now for automatically detecting the presence of these cancer cells if they can be monitored by such a contrast agent. And so I think combining all of these three concepts together is really where we are today. We're at the forefront of this technology, and I think Imagion is really entering this at the right time and providing the right type of agent for us to be able to probe this cancer.
Absolutely. It's incredible work. I'll maybe just go to you, Ward, and just let us know. So what was the purpose of the recent study at Wayne State, and why was it so important for the Magsense roadmap?
Yeah. So first of all, I think setting the stage of why we went to Wayne is that Dr. Haacke is just one of the absolute top minds in the world of MRI. Everything I know about MRI, I learned from him over several years working together. So when it came time to take on this study, there was only really one place in my mind that we were going to go because of a lot of the work that he and his team have done in improving these sequences, developing a lot of really advanced quantitative sequences, and being able to see things you couldn't really otherwise see with MR. So in doing that, we knew we had a few things that we wanted to address in the phase 2. First of all was reducing the dose of the Magsense agent. We knew from some of the work we had done in the phase 1 that one, that we should be able to reduce the dose considerably, and then two, that there was a big benefit to it. One was that it's going to reduce our cost, first of all, of per application of the agent, but two, also going to significantly improve patient comfort, safety, tolerability. So I think lead to an overall more successful study. But to do that, we really needed to substantiate those claims for the IND submission. So that's what we were really looking to study: is how low of a dose could we get to and still have a discernible, detectable signal. And I think we certainly have gotten there and have what we need to substantiate that and include that in the IND submission. But on top of that, I think the other thing for me personally, maybe, but also for all the radiologists out there, was really improving the image quality that we had, improving the sensitivity and readability of the sequences that we were using. That was one of the first things when I came into the company. Was one, I was incredibly excited about what they were doing from an MR standpoint, but saw a lot of room for improvement by using a lot of these advanced sequences that Dr. Haacke and his team and others around have developed and perfected. So that was something else we've explored in the study, and has given us a lot of potential for future directions and ways that we may be able to apply that. And then third was really looking at the application of quantitative imaging techniques into our protocol. And that's something that I think is going to lead to some really interesting results in the future, is rather than just looking at an image on the screen, now we're able to actually measure what is there on a pixel by pixel basis. We're able to evaluate these images much differently, track changes over time, but also provide a lot more certainty into what we're seeing. And there's a lot of different ways that we can apply that going forward, looking at potentially reducing the need for a pre-dose scan, looking at the development of various AI and automatic detection tools. So That may not be something that we use right away, but is setting the stage for a lot of exciting things that we plan to address in the future.
That's fantastic. Just over to you, Mark. So, what were some of the key findings of the study, and are you able to reduce the dose as was intended?
Uh, yes, I think we have successfully got this down to, as I mentioned, about 1 microgram per milliliter. This method we use, it's called a gradient echo 3-dimensional scan. And the advantage of that is that we can run this with very high resolution on the order of a cubic millimeter in terms of voxel size. So that's quite small, and that means that if you have some inhomogeneous distribution in the lymph nodes of the cancer, for example, that you have the ability to really be able to study this fine structure, because this may not be some uniform distribution inside the lymph node. So the methodology that we're using is quite flexible. We can change the coverage we have. We can potentially scan the entire breast, for example, or the upper area where the lymph nodes are, probably in just a few minutes with that type of resolution. And so this offers, as Ward said, if we run the scan twice, we're able to develop a lot of quantitative information at the level of 1 cubic millimeter. And at that point, the iron changes the tissue properties in a way that something called an R2 star map can be used to actually automatically detect these changes relative to surrounding tissue. So the technology is there, the speed is there, the comfort for the patient is there, and the advantage of the high field scanners is the signal to noise and contrast to noise are available to make this detection really possible.
It's all sounding very positive. I might just open this one up to the room a bit. So, just tell us, what are some of the implications of introducing quantitative MRI into the protocol, and what does this enable?
I'll go and give you maybe my analysis or the assessment of this, and hopefully Dr. Haacke can correct me if anywhere I may be out of line. But I think there's a couple really interesting directions we can go. First is eliminating that pre-dose scan. Right now, we scan the patient, then administer the Magsense injection, and then at some interval later, which is something we're going to try to optimize more in the phase 2 study, we would then do a second scan, and the radiologists are kind of looking for the difference between pre and post dose. It's just a fairly standard way to look at contrast MRI. However, by using the quantitative imaging and being able to sort of capture that unique quantitative signature, we may be able to completely eliminate the need for that pre-dose scan. That's something that we'll be looking at in future directions as well, in future work. And that is hugely important. One, for just the patient, you don't have to have 2 MRI sessions, especially if it's over a long time period between the pre and post dose. Maybe that even could be overnight in some cases. You eliminate that hassle for the patient themselves. It also makes it much easier from a workflow perspective for the imaging center, because they're only having to run one scan. You're not having to worry about timing. So I think that's going to be a really important direction for us as we look at clinical adoption. Not to say that otherwise that it's overly burdensome, especially compared to some other diagnostic techniques. But everything we do to make this faster and easier for the patient and for the clinicians is going to be really important. Beyond that, I think there are some other really interesting things that we may be able to do as we look at automatic detection and potentially like algorithmic and AI-based detection. Radiologists are incredibly busy. They're reading a tremendous number of scans each day. And anything we can do to make that job easier and faster and more accurate for them is going to be a huge help. I think it also, though, beyond making it faster, I think makes it more accurate and expands access beyond traditional academic medical centers. We had in our phase 1, we had our radiologists, our reading panel, look at these images. We had them draw little pictures of it and see what they're saying. And we had good concordance across the board. Everybody was seeing the same thing. They saw the same ratio of signal change. But those are with people who are academic radiologists. They're specialized in this area. There are a lot of patients who are beyond the reaches of those types of systems. And anything that we can do to put this in the hands of more people, to a broader audience and open up the access, I think is going to be incredibly important. And then outside of that, I think what we'll be able to look at in terms of analyzing the progression of the disease or staging the disease, which really is our primary focus, can be assisted by that algorithmic approach of looking at different patterns of the uptake into the lymph nodes, seeing how the progression of the disease is spreading. Those are all things that we can start to monitor with quantitative imaging and with AI. So I think that's incredibly exciting. And by implementing these sequences now ahead of the phase 2, we'll come out of that with a wealth of data that we can then start to train those models on. There's some other things I think just related to, I mean, I could go on around the current limitations of AI and radiology. It seems like everybody is so hot to trot about this subject and thinks it's going to solve everything, but there's just still some fundamental limitations of what you can do, whether it's starting to build big enough data sets. You still have a lot of subjective kind of human influence in the rating of the data sets and building these data sets. Quantitative imaging really starts to eliminate a lot of that. So it's pretty exciting. I think where we will be able to go in the future.
What were your thoughts there, Mark?
Well, I agree with what Ward has said. I think the interesting thing about the qMRI and this particular measure called R2 star is that one can potentially, especially in terms of following up treatment response, one can do a direct comparison rather than just a simple visual comparison. Oh, it's there, it's not there. One can look at this and see if this R2 star parameter has changed. And if that parameter is very high, it usually means there's a lot of cancer present. If that parameter starts to reduce, we can begin to say how effective that treatment has been. So I agree with Ward. And the other thing is that as we learn about that part of the body, Ward, that we know and map out the R2 star values throughout all the normal tissue there, then we can have a direct map of what is considered normal and what is considered abnormal. And so that qMRI image can easily be fed through AI, and AI can maybe do even more than just say, oh, yes, there's an indication of cancer cells there. It can say, here's what's happened from the initial scan to the second scan, and there appears to be some positive response to the treatment. So the AI can really make the analysis easier. And also, I think AI can help in the sense that it's not just the time involved for the radiologist to look at this. If we're talking about really small structures, it's possible the radiologist could miss those as they're looking through hundreds of images, or they're tired late at night. You have a few little bright spots in there. You might just think your eyes are tired with those bright spots. So in this particular case, AI, I think, has the potential to know where to look, what to look for, and how to assess it. And that's really training it, like a radiologist who never gets tired.
That's a really important point there, Mark, about looking at these smaller structures. And this is tied to some of the research work that we've done in the past, is that we've seen that by using this targeted imaging agent, we've been able to detect affected nodes that are tumors that are up to 10x smaller than you would with traditional CT or MR alone. And I think that just leads to that point, is that you can, not only can we tag them and find them, but now with AI we can light them up and make it easy and quick to find, and hopefully reduce any potential errors there may be.
Yeah, it's really interesting. Obviously, there's a lot of biotechs, that are trying to bring maybe new drugs or new technology to market, whereas this is more just growing and expanding on technology that's already there, but can always get better and more accurate. And it really sounds like it's setting a bit of a foundation for the future of kind of MRI and AI and automated detection, which, as you guys have mentioned, these are humans looking at these images. They're going to make mistakes, so obviously it'll eliminate some human error, I would imagine.
In eliminating the need for things like confirmatory biopsies. I mean, right now, conventional MR, you still, even with the amazing resolution that you have with it, you still need to go in and confirm with a biopsy. Uh, and that's what we're hoping to solve with this molecular MRI, is being able to provide that sort of precision and diagnostic information to hopefully avoid a lot of these unnecessary um, and painful, uncomfortable biopsies that patients have.
Absolutely. Maybe just lastly, Ward, for any investors that might be tuning in here from a business sense, and maybe you can just give them a little bit of an update on how the IND application is progressing, and maybe where to next for IBX and Imagion.
Yeah. So from a IND standpoint, everything is on track as we have planned and promised to the market. We are on track to submit, by the end of the year. This is an important step towards that goal, providing a lot of the good kind of confirmatory information about some of the things that we're doing on the imaging side. Outside of that, we've completed the manufacturing of our clinical lot of material. We're now in the analytical testing stage of that. We expect that to be released from the stability testing fairly soon. And after that, we should be fairly ready to submit. We're completing our, finalizing our study protocol right now, looking at site engagement. So we're pretty well on track from the standpoint of getting to our phase 2.
That's great news. It's good to hear. Well, thanks, guys, for joining us today. Thank you, Mark, for joining us. And I hope these study results come back positive and we get that application passed.
Absolutely. Thank you,
Very
Matthew.
promising.
Thank you, Dr. Haacke. Appreciate it.